This is a production of Cornell University.
So I prepared more slides than what I was allowed. So I'm going to, my introduction is right here. Many of you know me already. But my lab works on plant metabolism. The first question here is why we are interested in plant metabolism. And the fact that plant metabolites themselves find a variety of uses in human society, right? But plants don't make these compounds, these metabolites, for human consumption or human uh, interest. So they are produced mainly for various plant processes. Everything in plant growth and reproduction and development, stress response is finally impacted by metabolites. And so this is what fascinates me quite a bit, that we can go not only to the fundamental aspect side, but also to the applied side when studying plant metabolism. So all of this function is possible because plant metabolites are huge. There's like over 1 million metabolites predicted across the plant kingdom, produced because of the complexity of the underlying metabolic pathways. So the different questions in our lab are framed around 3 common areas. One is what is the scale of this plant metabolic diversity? It's surprising, but only less than about 2 or 3% of the plant metabolites can be identified. So second question is, what are the biochemical and the evolutionary mechanisms generating this diversity, and how can we use it for different human activities? We have worked with several model systems, including sweet potatoes, which are part of the morning glory family, tomatoes and Solanaceae family species, grasses, and some comparative genomics projects across plants. We do both computational and wet lab research, as you'll see later. So I'm going to talk about 2 project areas, just give a very quick overview of what we are working on. One is sweet potato and morning glory research, which several people in the lab have contributed. These are the ex-members and current members who are working on sweet potato. So why sweet potato? It's not really an important crop in New York State, but my history with sweet potatoes and morning glories, that my postdoctoral work was in acyl sugars in the Solanaceae family, and these acyl sugars don't exist in the sister Convolvulaceae or morning glory family, but they have different types of acyl sugars known as resin glycosides. The structures are different, and the acyl chains that are attached to the sugars which are in the black, those are different. This is a monitor. So what we found out in my lab is that we have a huge diversity of resin glycosides across 30 different species of the morning glory family. And this diversity allows us to do more mechanistic investigations of how it arose. So resin glycosides are important for sweet potato defense. So sweet potatoes that produce more resin glycosides are more resistant to nematode damage. And they're also medicinally important in Indian and Mexican traditional medicines. So we are interested in figuring out what are the enzymes that produce these resin glycosides. For that, using the diversity, we found out that most species produce resin glycosides in both, or accumulate in both roots and leaves, but only in this Dichondra argentea it's produced in the roots. So we did RNA-seq of these two, but these two are evolutionarily very distantly related. So we are also looking at a time series between the days after germination of the seedlings of Dichondra, and we see a significant difference in accumulation patterns, which we are also using for figuring out which enzymes are upregulated at different stages. We are also screening a bunch of different sweet potato varieties, 55 cultivars from the Sweet Potato Germplasm Center, and looking at resin glycoside levels and how they are associated with known insect damage patterns. Yes. Now looking at this diversity, we started to think of additional problems that could be answered. One thing that is commonly known is sweet potatoes are mostly produced in the South. North Carolina is the biggest producer of sweet potatoes in the United States. It's a warm weather crop, not suited for growth in New York. But there are many northern states that are interested in growing sweet potatoes. Cornell Cooperative Extension has a sweet potato program. we have variety trials that go on in different parts of the state, etc. So how do we optimize the growth of sweet potatoes in New York State? Because there are some challenges, as we know, cold temperature, but there is global warming happening, which results in a more warmer temperatures up north. At the same time, we don't have any problems with the pests and pathogens of sweet potatoes in New York State. So all the farming here of sweet potatoes is organic, which it, uh, produces a premium value for, if you're using these sweet potatoes, the colored sweet potatoes like purple ones for health drinks or dyes extraction, etc. It's a significant premium for organic cultivation. Um, what we found out through previous studies is that the concentration of anthocyanins and flavonoids in these purple flesh sweet potatoes is comparable to if the sweet potatoes were grown in the south. Um, we also found that there are over 250 anthocyanin and flavonoid signals in these purple sweet potatoes, which computationally analyzed produces a lot more signal than just using other studies. But there is a huge variability in how they are grown. So this is sweet potatoes started from the same slips, one in Geneva and one in near Albany, and this is the difference in the roots and the two different varieties. The interesting part is in the second year, this pattern was switched between the two. So we don't know what exactly is causing these differences. I mean, environmental factors have a significant influence on sweet potato growth, and so we have to optimize this through variety trials and through understanding the mechanism of root storage. We're also looking at arbuscular mycorrhizal fungi as a way to increase the robustness of growth patterns. These are important for phosphate deficiency, but they are also, these associations are important for increased drought tolerance, salt tolerance, and we are looking at whether they provide benefit against cold tolerance as well as drought and waterlogging, etc. And whether this colonization associated with flesh color. These are some preliminary results from our analysis with two different varieties, which shows that water definitely has a significant effect on sweet potato growth. But we found out that the only trait affected because of AMF is root biomass. All other traits are highly variable, but root biomass is significantly affected because of AMF application. And we are looking at the molecular and metabolic basis of this, of these differences. Okay. The second area that my lab is working on is very on the fundamental side of research, and this deals with the issue that there are so many genomes now available. The quality of the genomes is high. We can easily sequence diploid plant genomes. But if you look at how these genes are annotated in these genomes, the annotation is very poor. We find some genes using RNA-seq or QTL and GWAS, but we have to rely on annotations obtained from Arabidopsis for this reason. So only in Arabidopsis are experimental annotations high, but in all other species, the amount of experimental annotations from experiments is very, very low or non-existent. So we are working with various different approaches, including curation of existing research papers, phylogenomics, cheminformatics, and more recently machine learning to answer these questions. The eventual outcome of this research is that if we have better substrate annotation, if we can say, okay, this enzyme uses these substrates computationally with a high degree of accuracy, we can apply those annotation workflows to over 300 plant genomes, high quality plant genomes, and improve the annotation for basically all researchers across the world for these enzyme families. We're targeting 10 families right now and depositing all of these activities in global databases like UniProt and Gene Ontology. So there are Nate and Shinyu are working on this project. I think that's it. You might have time for a question or two.
Yeah, one quick question.
I think I overcompensated for extra slides.
This might take more than a minute, but how do you make sure the gene annotations are actually in the protein?
Yeah. So we are doing both computational prediction and validation using transient silencing, CRISPR, in vitro enzyme assays, etc.
All right, so I'm an assistant professor. My name is Lori Huberman. I'm an assistant professor in the plant pathology and plant microbiology section. And my lab works on characterizing the genetics of how fungi sense their environment. So these guys don't work. Okay. So we're the School of Integrative Plant Sciences. So why do we care about fungi? Well, fungi are an integral part of the plant ecosystem. They're really important for nutrient cycling in the environment. Without fungi, we'd just be like up to our ears in plant biomass. And they're also really important, as Gaurav was saying, for forming symbioses with plants and helping plants uptake nutrients. Unfortunately, fungi are also pretty serious pathogens. So for humans, they cause about 1 billion infections a year and cause 1.6 million deaths. They're pretty serious animal pathogens, currently causing extinction or threatening a number of species in bats, amphibians, and reptiles with extinction. And then for plants, they cause about a 10 to 30% reduction in crop yield because of fungal diseases. And so it's really important that we understand the basic biology of fungi so that we can better figure out how to live with them and, take advantage of their good traits and avoid their bad traits. So what my lab is really interested in understanding is how fungi interact with their environment. And so fungi can sense and respond to their environment in a number of ways, but one of the signals that they use is chemicals. And so these chemicals can tell them if there's another microbe around, if there's another fungus around to mate with or fuse with, if there's any food around, if there are any plants around to either infect or form symbiotic relationships with. And the main overarching question that really defines my lab is how do cells integrate environmental chemical signals and make decisions? And so one of the big questions that my lab looks at is how fungal cells sense and respond to nutrients. And this is really important for a number of reasons. So for pathogenesis, it's known that nutrient sensing is really important in pathogenesis. So if we think about pathogenesis of plants, one of the main defenses plants have against microbes is a really tough cell wall. And fungi are able to produce and secrete enzymes that break that plant cell wall down. And the regulation of these plant cell wall degrading enzymes is regulated by the fungal cell's ability to sense and respond to nutrients. And then it's also been shown that the ability of fungi to distinguish between preferred and non-preferred nutrients in their environment is associated with virulence. So if you eliminate their ability to distinguish between preferred and non-preferred nutrients, they're not as able to infect plants and animals. Fungi are also really important in industrial production of enzymes and biofuels and other bioproducts. And if we want to do this in a sustainable way, the sort of generally accepted idea is that we would take plant biomass and then break it down into its component sugars using enzymes produced by filamentous fungi. And then these component sugars can then be fed to engineered microorganisms that have been engineered using metabolic engineering to produce biofuels and other high-value products. And so my lab is really interested in understanding how these plant cell wall degrading enzymes are regulated from filamentous fungi, and also how fungi are regulating the utilization of plant-derived carbohydrates, and then how that is regulated as it goes through to producing the precursors that people use to produce the engineered products. And so one of the big questions that my lab is really interested in answering is how do cells know what they're eating? And so if we zoom into the fungal cell at the molecular level, fungi don't have eyes or tongues or mouths, and so they have to sense their nutrients using receptors and transporters. And so at the cell surface, there'll either be a receptor that can bind to a nutrient or a transporter that can import that nutrient. And then either signals from the receptor or perhaps the nutrient itself will act to signal through a set of signaling pathways that goes on to activate the transcription of transcription factors or to help to transition a transcription factor from an inactive to an active state. And then those transcription factors go on to activate all of the genes that are necessary to utilize that nutrient. So these might be secreted enzyme genes that break an insoluble nutrient down into soluble substrates, catabolic genes, and transporter genes. In the lab, we frequently will expose a fungus to a single nutrient at a time, but that's not what it's going to experience in the wild. And so in the wild, fungi have different nutrients that are at different levels of preference. So the more energy that it takes a fungal cell to utilize a nutrient, the less preferred that nutrient is going to be. And so in the wild, you would expect to have a fungal cell experiencing a number of different nutrients, some of which are more and some of which are less preferred. And so if there's a preferred nutrient around, then that preferred nutrient will be sensed by a different set of receptors or imported by a different set of transporters. And then these will signal through a different set of signaling pathways to a different set of transcription factors, which will repress all of the genes necessary to utilize that non-preferred nutrient source when the preferred nutrient source is present. And so my lab is really interested in identifying and characterizing all of the genes that are involved in sensing the nutrients, the signaling pathways that are involved in transmitting this signal, and the transcription factors that are involved in activating or repressing the transcription of the genes necessary to utilize these nutrient sources. And so we go about sensing or identifying and characterizing these genes in a number of ways with different genetic screens and high throughput sequencing. But one of the advantages to working in a microbe as opposed to a plant is that we have a lot of powerful tools that we can use to do pooled screening. And so I want to tell you about one of these. And so we can do high throughput functional genomics using massively parallel screens. And so we have a library that was produced, that was made by a collaborator at UC Berkeley that has about 300,000 mutants in it in a Basidiomycete yeast strain that we work with. And so each of these mutants is tagged with a unique DNA barcode, and each of those DNA barcodes is associated with a disruption in a particular gene. And so we can take our pooled library, grow it up, and then expose it to whatever experimental condition we're interested in. Most mutants don't really have a phenotype under most conditions, and so most of our mutants are going to grow at about the same rate. But some of our mutants are not going to be able to grow well at all in whatever our experimental condition is. And so these mutants will be underrepresented as compared to the rest of the population. And when we analyze the barcode abundance, we will see a decrease in barcode abundance from the starting population to the final population. And we can infer that whatever gene is disrupted in this mutant is required for growth under this condition. Other mutants, like this yellow mutant here, will be growing much more rapidly than the rest of the population. And so these cells will be overrepresented as compared to the rest of the population. And we can infer that whatever gene is disrupted in this mutant is detrimental for growth under this condition. So I just want to give you a really quick example of how we can use this in the lab. So we did an experiment where we took the library, we grew it up in rich medium, and then shifted it to medium in which either glucose was the sole carbon source or cellobiose, which is a breakdown product of the plant cell wall, carbohydrate, cellulose was the sole carbon source. And then we looked to see what strains were either over or underrepresented as compared to the rest of the population. And so we found strains that had disruptions in a transcription factor. And so when we made a clean deletion of this transcription factor, which we named CBR1 for cellobiose regulator 1, we see that when we grow it in media containing glucose as the sole carbon source, it's able to grow just as well as wild type. But when we grow it with cellobiose as the sole carbon source, it's completely dead. So this transcription factor is indeed required for cellobiose utilization. So what I've been talking about so far is doing these kinds of experiments in yeast, which are sort of like normal microorganisms. So when they go through mitosis, they also go through cytokinesis. And so for every nuclear division, you get a cellular division. Filamentous fungi are not so convenient in that way. And filamentous fungi are about 99% of the fungal species that exist in the world. And so what you're looking at here in this, the quality really dropped. Anyway, whatever. What you're looking at here is a fungal colony. So this is a filamentous fungus. So what you're looking, these are fungal cells. And what's zooming around in here are fluorescently labeled nuclei. And so if you think about doing genetics in a kind of organism in which nuclei are sort of free flowing throughout the population, you can imagine that it becomes really challenging. And so one of the things that my lab is interested in doing is transferring the technology that this group developed in yeast to do these barcoded mutant libraries and making barcoded mutant libraries in filamentous fungi, which we're currently doing. And so in this case, instead of looking for a change in the abundance of mutant cells, we'll be looking for a change in the abundance of mutant nuclei. And so this is what we're working on now. And so with that, I'd like to thank the members of my lab and our funding sources, and I'd be happy to take any questions.
All right. Good afternoon, everyone. My name is Vipan Kumar. I'm an associate professor in weed science at soil and crop science sections. It's been 1 year, actually today, being in Cornell now. So I'm going to share some of the things we have started with my program. My appointment is 60% research and 40% extension. So I meet and talk with a lot of stakeholders, especially producers. So I'm going to show some of the applied research we have started in Cornell over the last year. So just 4 major things or aspects of my program areas I have outlined here in the slide. And you can see the monitoring new weed species and distribution of herbicide-resistant weeds. This is a kind of my core emphasis of understanding what's going on in field crops in New York in terms of herbicide-resistant weeds. What are the new species we are seeing in our regions due to changing climate or other management practices? The second aspect is evaluating herbicide programs. What are the alternative effective options do we have if we have resistant populations in the state? Third aspect is looking at the precision weed control. What are the new tools coming into the production system which we can use for site and species specific weed control in field crops? And the last but not the least, what are the alternative or non-chemical weed control options? If you're not going to use herbicides, what are the other non-chemical options do we have? So moving on to the new weed species, since 2019, New York State, we have seen 2 most problematic weed species have come into the state. Number 1 is the Palmer amaranth, and the second one is the water hemp. Both are pigweed species, highly problematic in Midwestern and Southeastern states. This is not a good news for the producers, especially for field crops. And majority of these population we are seeing having resistance issues, especially commonly used herbicide glyphosate resistance in these populations. Currently, this is the work being done with my colleagues, Dr. Lynn Sosnowski and other weed science colleagues. We are kind of monitoring where we have been seeing these populations, especially Palmer and water hemp. Right now, currently, we have 6 counties where we have identified Palmer amaranth population in the state. However, for water hemp, we have 16 counties where we have identified water hemp population since 2019. Coming to Cornell last year, I was very excited about some of the things going on in the field. We collected some water hemp populations from the grower field, especially in central New York, in soybean fields. And the idea was to see what's going on, what's the response of those population, especially to commonly used herbicides, things like Roundup, Cobra, Callisto. These are commonly used herbicides in soybean and corn. We made clones out of these collected plants, as you see in the greenhouse, 4 populations. We cloned them and we sprayed them with the 2x rate of each of those herbicides using our spray chamber here in Ithaca. What we found, 4 of those populations, water hemp populations, survived glyphosate 21 days after treatment. And even they produced seeds. We collected seeds out of those plants later in the fall last year. So moving forward, what my program is looking at, we are collecting weed seeds, especially these water hemps, common water hemps, horseweed, foxtail, common ragweed, and velvet leaves across these field crops, and trying to characterize or see the distribution of herbicide resistant or multiple herbicide resistance in these populations across the state. Because if we know what is the distribution, we can plan or develop programs to manage those populations in those different situations. In terms of other new species, I have came to know we have Johnson grass in counties in southern New York, very limited, confined areas we have identified Johnson grass population. We have also come to know like kochia, which is a tumbleweed, weed of the West. This is the most troublesome weed in the Western US, has also been identified one population in Western New York. So these populations are on the horizon, or new species are on horizon, and these are highly, highly invasive and prone to develop herbicide resistance. So we are keeping our eyes open and trying to educate our folks to look for those population and be proactive. The second aspect, as I mentioned, I'm not going to go too much details. We are evaluating herbicide programs because some of the questions come from stakeholders. What are the options if you have a Roundup-resistant water hemp? How are we going to control it? What are the options? So this is the work we started with the farmer in central New York last year. These are the treatment, messy slide, but just stick with me. This is different programs we are looking at. We have a pre-applied programs and by post-applied program, and then just post-applied programs. Different herbicide mode of actions, different chemistries, different premixes, different rates. So the idea is to look at what are the approaches or strategies we can recommend to our folks who are dealing with the glyphosate-resistant water hemp or palmer amaranth or horseweed. Preliminary results, this is the 1-year data from a grower field, pre-applied herbicides. I'm not going to go too much into the details what those are, but look at this. This is just a pre-applied, nothing been done, just a one shot of pre-herbicide application. And glyphosate-resistant water hemp, this was the highly infested field in soybean. Very inconsistent control, visual control we observed throughout the season, especially for glyphosate-resistant water hemp. But where we had pre by post options, we had better control throughout the season if you look at those 3 ratings for glyphosate-resistant water hemp. So we are trying to repeat these experiments and kind of educating our stakeholders like what options they have to pick if they have a problem like glyphosate-resistant weed population. Third aspect, very briefly, precision ag weed control or precision ag tools. What are the tools available and what we are looking at it? We are trying to look at the sensors and new cameras, especially OAK-D cameras, trying to quantify the weed densities as well as the weed biomass. We are trying to automate that system so that we don't have to go and destruct the weeds in the crop. This is a multi-state project. We are involved with the GROW team. And if you know GROW, getting rid of weed, it's a USDA-led team out of Maryland. So this is the one project we are looking at with the different partners, different states. And you also see one of my graduate students is also looking at the detection of palmer amaranth as well as water hemp in crops like soybean and corn using machine learning algorithms. Along with those precision ag tools or machine learning algorithms, we are also looking at the impact of precision ag technologies like nozzle types, spray volumes, how they are impacting some of our chemistries for these weed control. Moving on to another tool we are very excited about, and I really appreciate Cornell Ag Experiment Station giving us the support to buy this new tool. This is called Harvest Weed Seed Control or Ready Crop Seed Control Unit. So what it does is basically it collects the crop residue when you are harvesting crop. There are also weed seeds there, right? So it collects those weed seeds and destroy those weed seeds basically. I have worked with this unit at Kansas State, and we have looked at palmer amaranth seed destructions. We have found more than 90% of seed destruction with this tool if we use this with the combine. And this is the new combine we have bought at Musgrave Research Farm. And this is the unit, Ready Crop Seed Control Unit on the back of the combine, which pulverize or destroy the crop residue as well as the weed seeds. And this is one of that impact mill. There are 2 impact mills kind of crushing those seeds. So going forward, we are looking forward and excited projects looking at integrating these tools with other kind of ecological-based weed control tactics like a cover crops, as well as a chemical as a system approach, many little hammers, how they can help to control or manage the weed seed banks in our field crops. That's all I have. Thank you.
Good afternoon, everyone. My name is Jiang, assistant professor in Horticulture Section within SIPS. So I joined Cornell back to 2019 as assistant research professor, and then changed my kind of trajectory with more appointment on the extension, because we identify not only we need to develop these new technologies, but the adoption or commercialization of this technology is also vital, not only for research, but also for the production side. So today I'm gonna just talk about some of the ongoing projects within my team, so hopefully can make more connections with the plant breeding and the genetics section. So I established a lab short for Cyber Agricultural Intelligence and Robotics Lab, short for CARE. So it reflects the two things. One is what I'm doing every day. It's basically multimodal sensing, robots, and then artificial intelligence, but with more a dedicated focus on the agriculture applications or the food applications. And second, I think CARE is the best word I can describe myself and my lab mission, that as engineers, we really care about our human society, which I'm very proud of. And of course, from my lab, we use a little bit different approach. Instead of tackling one single aspect and really get everything solved very, very deep, we take the systems engineering approach. So that means when we try to optimize the entire system, we think of all the possible ways we can improve, and then as a whole collectively, we can achieve the goal more effectively and also fulfill our near-term and long-term goals. I just really want to show this thing, and my program focuses on specialty crops, and I think they are very important to New York State, economy. But as a person, I think that's something I eat every single day. I love to get them and really make this whole industry more successful in the future, because that's really the food supply the rest of the world. So back to the overall concept, I kind of introduced this concept called one egg. So as we consider the systems engineering, very important for us to first identify what are the key agents there. And in agri-food system, we usually have two, right? One is the people, ourselves, either as a researcher or grower, or even just a public person to purchase something. That's our interaction started with agriculture and food. And then of course the agri system itself, starting from breeding, and then in season, and then during the season management, and then after season for the post-harvest evaluation. So in the past several thousand years, we rely on our direct interaction to get experience, and now with more and more tools, we hope to use a more data-driven approach to summarize our experiments and find the new knowledge that we can translate to the next generation, and we continue this whole cycle. But for me is whether or not we can more systematically add this technology layer. So instead of let the human directly interact with the agri-food system, we create a universal interface between these two agents, so we can more systematically understand all our findings, and hopefully in the future we can use artificial intelligence system to unify all the findings and really push the progress for designing the better agri-food system in the near future. So the first thing I want to share is actually the bird ecosystem. I believe from my colleagues from Cornell Agritech, you heard a lot about Blackbird. It's a kind of a microscopic imaging robot. We designed that for grape powdery and downy mildew to replace the traditional human observation under dissect microscopes. And now with each of these Blackbird, we can replace 60 human hours, right, as an equivalent. So we kind of test that on a very wide range of possible applications, not only focusing on the leaf tissues, but in recent years we test them on insects. And just in last year, we worked with someone from entomology to use this robot scan the entire pest sticky card. So that's the kind of lower two corners you see. That's a seed corn maggots, and they try to use the microscopic information to reveal what's their genders, so that they can calculate the ratio and reveal some questions very important from the entomology side. So we are very excited about these applications, and that's how I see we offer new instrumentation for many of the scientific programs here, try to better understand the science. And of course, we are kind of making progress to convert the traditional, blackbirds with RGB microscopic imaging capacity with more like a hyperspectral thermal imaging. And those kind of being used for various, interesting applications. And we'll also welcome for you to contact us and get us some cool samples that we can start to, explore the cool trajectory on some of the scientific questions we are interested in. So the other part is getting out of the lab, right? That's where agriculture, to me as an engineer, is really, really attractive, is the complexity. Once we get into the field, everything can happen. And there is no equation that I can put as a function to calculate what's the final results. So that's the reason, in addition to those lab robots, we also design the field robots to acquire the information so that we can evaluate the performance of the crops in the field. And then we can tie them with the environmental measurements and better understand how these interactions really occur. Especially, I think that's the interesting points for many of the people from plant breeding and genetics side is the field, based high throughput phenotyping. And that's exactly our answer, is to use robot to replace the, human power for more objective and accurate evaluation in the field. So these are just some videos we took from various places. In New York, we tested for grape pottery and the downy mildew. And in California, we tested for grape leaf roll virus, and we got pretty high accuracy, almost over 95%. And at the right corner, you see that's the final map. Each actually stripe represents a specific germplasm in a population, and the color indicates their disease severity. So red means highly infected, and more white or grainer means they are healthy. So with that information, I hope my collaborators from this breeding program will tell us what the genes or QPLs associated with the disease resistance, so we can better integrate them into the future breeding program. So with all this information, so what we kind of working in progress is called a real to sim, sim to real closed loop system for digital twin. So digital twin is a concept proposed by NASA about 30 years ago. The whole aim is to foster the production development in the digital system, so that later on, when they get into the physical manufacturing process, everything is already there, so they don't need to go multiple rounds for the physical development. So similar idea here now for plant breeding or plant science in general is we want to use all these robots getting the real world data, and then we can design some AI systems to reveal what data really tells us, so we can form this kind of a digital twin with the physical system. So in this digital twin, one of the kind of key questions I would love to hear some feedback from the breeding and genetics section is how we can really inject the genetic information, the environmental information, to reveal the G by E effects during this simulation. Because that's where we really have the opportunity to move time forward, how these crops are gonna look like after 20 years of the development, and especially given these unprecedented environment conditions. And that will give us a lot of information about what we should prepare for the future, rather than passively waiting for, oh yeah, that's the time we need to start something, but that's already too late. And at the same time, we use this simulating environment as a test bed for us as roboticists to design new robots. Because these simulated environments largely incorporate all the information from real world, the robots designed there will likely just behave regularly as compared with we tested in the field season. But now in this digital world, we have multiple times to do this iteration without any of the waiting time, right? Because usually that's the key challenge for agriculture. I have to ask my student to wait a full year to test the next round of the robot development. But now with this digital system, we can test that anytime we want, as long as we can really make it more realistic. And then this data can be served as another training set, getting into the model to better serve the real world data, like how this tree's architecture look like, how I need to prune them and really form a better world in the future. And of course, there are some like cool 3D models. I don't know how to really mathematically describe their interaction there, but I hope that's the connection here. I can, work with you all about that. So ultimately, we want to put everything together, across the scale. In the past, in my opinion, it's more like we find the knowledge at the individual scale without much, connection based on the data-driven approaches. And with this kind of a whole system revolution, we hope to now connect all these dots together. And later on, we can use an AI system to unify all the findings across different spatial scales and transfer the knowledge back and forth. So with that, I actually gonna skip my extension because maybe that's something we can discuss along the time. But the key thing I really want to make it happen is I'm willing to hear the feedback from breeding and the genetics side, how we can incorporate other considerations like the society factor, the financial sector, the healthcare sector during the breeding process rather than the current breeding trees we are interested in. With that, I just stop here. And if you have any questions, just feel free to drop me an email. Thanks.
Okay, thank you so much. We have a few questions on the line. Okay.
Oh, for me? No.
Let's start with first one from Anne. Chance we could do this and cell. So this one. For me? Okay. Yeah, right. Oh, all speakers come forward.
It could theoretically be possible with plant cells. We'd have to do it, I guess, with plant. I mean, I'd be definitely happy to chat with you about it. There are some difficulties just in the fact that plants aren't haploid, and also it would have to be in cell culture. So, and I don't know a lot about plant cell culture, but I'd be happy to chat with you if anyone was excited to try to move this technology into plants. I'd be happy to chat with whoever that was.
Good. So 2 comments from my group about seed destroyer. Posted also. No question, just awesome.
Oh, thank you. Thanks.
Also, the digital twin physical and the process based on models. So,
Oh yeah, that's a great question. So that's where, in my opinion, we need a lot of work. Because from my work, I largely gonna really push for the data-driven approaches because that's where I started. I gather all this data, I can design a model to reveal what data means. But I think that's the connection point and the collaboration point with many of you is how this data can be summarized as new knowledge to really push new mechanistic modeling. Because in my opinion, data-driven has some bias and it cannot be easily avoided. For example, the number of possible genotypes we tested here only represent something available so far and only available in whatever ways we can assess them, right? But not really reveal the whole population in the world or in the nature that we want to test them. And I would want more like a hybrid, way that combine both the principles from biology and also the data-driven approaches to do a kind of another closed loop between these 2 approaches so that we can do better modeling in the future.
how much does a seed destroyer cost?
Yeah, right now, for research purpose, we have discount, but for growers is about $80,000 to $85,000. The unit I have is a ReadyCop seed control unit, and there are other impact mills available, especially integrated harvest or Harrington seed destructor from Australia. There are some differences between the impact mills. It comes with the 1 impact mill versus 2. ReadyCop is the one who's building 2 impact mills, and that's the one I have experience with at Kansas State. So that's about 80,000, but we had a discount when we bought it as a part of a research aspect. So, thank you.
The room comments, questions from the room.
I have a question for Dr. Kumar. Are you or any of your collaborators looking at sequencing the glyphosate-resistant crops and weeds and trying to understand it?
Yeah, definitely. I missed to mention that we are hiring a graduate student myself, Dr. Sosnowski from Cornell Agri-Tech, who's going to work at characterizing these populations, especially water hemp and palmer amaranth. And the idea is to sequence those and understand the genetics and molecular mechanism. As a student, when I was in PhD, that's what I did. I like to have a student who kind of dig it out. What's going on in a population in New York versus in Midwest, if this has happened like a machinery movement, or this has happened with some, other means of movement, because this palmer or water hemp was not here before 2019. What's the genetic diversity can tell us among these populations? So some of those aspects, the student is going to look at it. Yes, thank you.
My question is also to you, Doctor.
Yes.
In the beginning, you were saying something about extension, and I think you had time constraints to explain what you're really doing with extensionists at the moment.
Yeah, when I mentioned I'm a 40-person extension appointment, that means part of my time I've been spending with my stakeholders, especially producers in field crops, helping them to control these weeds. I do travel different parts of the state. Tomorrow I'm going to Rochester, giving a talk at New York Soybean and Small Grain Conference. So basically 300 people or growers going to show up and ask all kinds of questions, what we are doing now. What herbicide is working for horseweed or mare's tail or water hemp? And they're going to have a very diligent questions on the future of weed control strategy, especially when herbicide resistance is getting more and more problematic. What options do we have? Those kind of questions. It's going to be very, very exciting and interesting discussions. And that's where I pick some of these ideas for my future research from those producers, what they're looking for and what should I do to help those guys.
Are you looking at organic measures already?
Yes. So those non-chemical tools, I just mentioned two of them, cover crops and harvest weed seed control. There are more tools are available now. Basically, electric weeder is another one we are going to look at it. And these tools we are not looking at as a silver bullets, like a single tools, but as a multi-tactic approach. Many little hammers, how they can come together in a system, in a crop rotation, help managing the seed banks. So we are targeting the seed banks of these resistant populations. So of course, organic folks are my stakeholders. Yes. Any other question?
For everybody, but for you, Jiang, I'm thinking of your wonderful microscopic robot. I'm just wondering if you could kind of give a hint about how hard it is to train the system to recognize whatever new phenomenon you're going after. So say I was looking for some microarthropods or some nematode, how much do I have to budget essentially, given you've got this genius system to get it to recognize what sort of an organism I've got under that well?
Yeah, thanks for the question. So very short answer, it's no longer being that expensive. If you have a student who are willing to spend some of the working hour, label some like several hundred images, I can guarantee pretty good accuracy, as long as your microscopic sample is not too crazy. For some of the past sticky card we worked, in last year, they would attract like over 500 passes there. And to be honest, this is the first time I saw the data with all the legs from one insect, being spread out anywhere that I can find. So with that kind of a situation, it's just really hard for anyone, right? Even for human. Let's just use a human as the baseline to talk about the performance. It's even hard for human to find this information than it's perhaps being more expensive to train the AI system. But otherwise, if you feel, okay, with some like a very reasonable training, my students or technician will be able to do. And now I would see it's going to be cheaper to train those AI systems for you to identify the interesting of patterns, within those images. So I think it is there, but of course, for specific applications, we might go some, moderate modification, trying to make sure it achieves the optimal performance there.
This has been a production of Cornell University, on the web at cornell.edu.